And I think, the major in this area will be precision medicine, where the things are happening on each individual, how we are looking at their all the molecules and trying to give them a better, kind of diagnosis for the disease, or looking at the better treatment, or even prediction for their potential, severity of the disease, right? So that's where, for the cancer and other kind of other health diseases, this technologies and multiomics is going to be very important, and started playing a lot of role already. We have to not look at blindly, but very comprehensively every individual and their signatures to be much more accurate and more precise. And personalized medicine is where things are going to grow further, and proteomics will play a very important role in that. Again, like, the COVID vaccine did not come overnight, right? It was the several decades of the research and decades of the investment which has led us to help out in that time, right? So likewise, I think there is a need for some bigger common infrastructures in the country, where then big projects like this, when you think about, thousands and lakhs of individual to be screened, individual lab and facilities like my cannot handle that kind of big initiatives alone. So we need to build those initiatives like China.
Thank you for joining Once Changing the World. My name is Eddie Avil, and I'm delighted and honored to have with me Dr. Sanjeeva Srivastava. He's a professor at IIT Bombay who leads the Proteomics Lab and is the founder of Proteomica International Private Limited, who's renowned for his groundbreaking research in proteomics, especially in cancer, brain tumors, and tropical diseases like malaria, with collaborations globally and leadership roles in organizations like the Human Brain Proteome Project. He also developed BrainProt, an integrative omics knowledge base for human brain diseases. His contributions have laid the foundation for India's emergence as a global leader in proteomics and life sciences, and he's titled the Proteomics Man of India. So, Doctor, really appreciate you taking time and being part of the podcast. Now, I have done a couple of episodes for genetic editing, so my audience has a clue of what the Human Genome Project is or was. I mean, it was completed in 2003, and we kind of mapped the entire human genetic code, and it's kind of unlocked the blueprint, yeah, or the source code of life, the DNA, and it's enabled genetic editing, early diagnostic, gene therapies, and so on and so forth. But there's not much understanding of proteomics, so it will be great if you could start with explaining, maybe, just to our audience, explain what proteins are, its role in a body, what proteomics is, its role in helping us understanding proteins better, and how has the proteomics field evolved since you started your research?
Great. Thank you, Eddie, first of all, for your kind invitation. And I'll just take maybe 2 decades back, when I was finishing my master's degree. That was the time when Human Genome Project was getting completed, and even I was very excited to that the whole scientific community was excited, and we are all feeling that all the secrets of human is going to be totally unraveled, right? And so that's the time when the draft human genome came out of the things, as well as many other model organisms got published. And while it turned out to be quite useful information, we got a lot of detail for the blueprint of life, but it did not turn out to be unraveling all the secrets, because it turned out to be we are not so different than monkey, chimpanzee, or even the other model organism like Drosophila, or mouse for that matter. So therefore, the quest of looking at new molecules and dynamic molecules of life is started. And just on the basic side, we have a central dogma of life, where DNA or all the genes, they do the transcription when all the RNAs are being formed, and from there the translation happens. All the proteins are being formed. So therefore, the genes and the genome that forms the blueprint and the very static, blueprint of the life. But then a lot of things happens and changes over the time, from the transcription with the RNA and the translation with the protein, and even the post-translation modification, which includes something as simple as just adding a phosphate to a protein, makes it very different molecule like phosphorylated protein, or a sugar moiety to the protein adds makes looks like glycosylated protein. So therefore, the proteins then they are the one which are driving all the function, and we can say that they are something which is looking at range of cellular functions, if the diseases are happening, they are actually results of the malfunction of these proteins. And therefore, a lot of focus from the pharma sector and the researcher and the drug industry is also looking at which proteins are actually altering the, and getting malfunction in the disease. Can we find out those and use them for therapeutic purpose, right? So, therefore, while the analogy is very simple, when we talk about genome, we are looking at all the genes, but in the proteome, we are looking at the set of all the proteins that are expressed by the genome. But when we think about from the basic four codes of ATGC for the genes to moving to the language of the proteins, which is 20 letters now, not 4 letters, 20 amino acids, it right away becomes very complicated. So therefore, what we thought, which we have really understood very easily, genome, it turned out to be quite complicated for the proteome to understand because of the diversity and the functional groups which are involved in making the proteins, right? And I'll just take simple other example. For example, uh, we all see the butterfly looks very beautiful, right? And if you go through the developmental stage of butterfly, process known as metamorphosis, where you see a caterpillar and then moving to different type of larva and different pupa stages to make the beautiful butterfly, the genome remains static, genome remains same in all the stages. But what makes it look so beautiful with the beautiful wing, essentially the proteins. So proteins at the end, as I said, like the dynamic player, which are giving us the ability to do a lot of function looking at the dynamic way, while genome gives the static and the basic information. But we can take it from there and go to transcription with the RNA and the translation with the protein. Therefore, the focus, as I said, after the genomics project, shifted toward the transcriptomics, which was RNA sequencing and looking at the RNA-based analysis, and followed by the proteomics, looking at all the proteins and the building blocks of life. So that was kind of my journey as well, because I felt very excited that I should now venture into the area which is totally unknown territory at that time. Uh, and then why not should try to contribute in that area? And I had at least some basic good biochemistry knowledge of proteins. So I thought to build further from it and now look at not one, two protein, but look at the entire set of the proteins of human and look into their role in different type of dynamic processes.
Doctor, thank you for explaining this beautiful. DNA, RNA is the source code of life. It's the proteins which is the dynamic workflow. And thank you for explaining this so beautifully. Now, your lab uses two-dimensional difference in gel electrophoresis as well as mass spectrometry and proteins microarray techniques to capture the and understand proteomics. Can you explain how all these techniques work, and is there like a personal favorite amongst these techniques which gives you the best result when you talking about like capturing and understanding proteins?
Let's say a lot of time we do research on cancer and we look at from a given tumor tissue of a cancer patient, what all proteins are changing as compared to the normal counterpart, right? So what our goal is, from the tissue sample we are looking at 10,000 protein, from the blood sample we are trying to look at 2,500 proteins. So our goal is to look at thousands at the scale of separation and their quantification. So then we are always looking at technologies which could try to first separate these many proteins and then quantify the proteins. So therefore, there are different type of principles are involved in the process of doing so. The first technique, which was quite a bit of important in the proteomics field, was known as the two-dimensional electrophoresis, which actually started even before I was born, and that was in 1975. That technology was done by two scientists, Close and Farrell, and they looked at proteins property of isoelectric point and their molecular weight, how to separate protein based on these properties, and look at in the gel the spots and try to then separate those and look at their intensity values for quantification. But the technologies are very rapidly progressing, and the goal is, and the race is towards looking at as many protein as possible, because from a given tube, if I have this tumor proteome sample from a given patient, using one technology, if I can separate 2,000 protein, and using another technology, I can separate 10,000 protein, why not go for 10,000 protein? One technology which has made the maximum impact, and currently is my favorite as well, is known as the LC-MS mass spectrometer. So this technology, um, here after you have prepared the protein sample, it try to ionize your peptides. So proteins are being digested, and those gets peptide form. Those peptides are then getting ionized. They move inside the mass spectrometer, and then they get further separated based on the mass and charge ratio. And then the different type of mass spectrometer, which try to, because now we are talking about thousands of protein and maybe millions of ions coming out of it. So how well we can separate in a very, very fast manner becomes, our goal for this particular area. So therefore, the different type of nut and bolts involved in the mass spec, like Orbitrap and quadruples and TIMS and the different type of ion trap, different type of mass analyzers are involved, which help to separate these ions based on different physical principles involved. So this LC-MS is currently the most important technology, and this is currently my favorite as well. Protein microarrays and the technology which I mentioned, that technology is essentially looking at very different dimension of adding the data analysis. For example, in the mass spec based work, we are looking at something very unknown, right? You're starting from your complex tumor sample or any biological sample and looking at screening all their proteins, thousands of proteins. While the microarray, you are starting with a chip, and that chip contains the proteins printed already, right? It means you can't add something new. Whatever knowledge of the proteins you have, that knowledge of protein you can add on the chip. But what you can do more on that is essentially when you talk about the complex sample like blood or the plasma of human, they are having very abundant protein, like those some protein like albumin and immunoglobulin, they're quite abundant. And then there are many of the important protein which are quite low abundant. Think about, like, most of us when we get any infection or any kind of disease, the blood test is happens as the first thing, right? Which is very important for the clinicians. So there's a lot of emphasis. Can we identify some markers from the blood sample? And because they are being tested routinely for all kind of diagnostics. So if we can think about having, let's say, plasma sample to look for biomarker of cancer or different diseases, we first need to get rid of those abundant proteins, right? So those albumin and IgG, you have to remove them. Then only low abundant protein will be seen in the instrument. So therefore, mass spectrometry has the limitation of separation in the 10 to the power 5 order of magnitude. Whereas in microarray, it can go to 10 to the power 12 even, 11, 12. So therefore, here we are just going to take undepleted sample. We are not removing any kind of abundant protein. We directly take the plasma sample, probe on the chip, and trying to utilize which kind of antigen going to bind from the antibodies present in our blood sample and looks for those kind of immune reactivity. So it looks at different chemistry, at different ways of looking at protein and their autoantibody level or the humoral immune responses. For those applications, protein microarray holds a special role. Mass spectrometry is the major workhorse which looks for almost everything, but with some limitation of the dynamic range. But that's where microarray fits into the place, and let's try to look at how best we can try to look at the all the dynamic low and high abundant protein at the same time. So with all the efforts of these technology, at least as I was talking, proteomics has really moved quite a bit. Like, still we are trying to catch up pace with the genomics, but like the genome project was completed in 2003, even draft came in 2001, and the first draft of the human proteome came in 2014. It took almost 13, 14 years to show the first draft of the human proteome, which was still only 70%. Now we have almost 93 or 94% of the human proteome mapped as of now. Still, there is a gap of those 7% protein, which is known as a missing protein. It means we are still trying to say how to detect those protein using technologies. So that's, as I said, one technology cannot answer to all our questions, and protein microarrays can give us some answer which sometimes is not possible using the mass spectrometer. But mass spectrometry currently remains one of the major workhorses in our field.
We are probing these proteins, obviously these are nanoscale, and understanding with these various tech stack, and like you said, I mean, and from 2014, which we had like maybe like mapped out 70% to right now it's like 93 or 94%, possibly maybe just around 7% of proteomics that we need to map further. I mean, how cool it would be when we are able to completely map and understand the proteomics in a way that then maybe that the game begins. I mean, so when possibly maybe in the near future we are able to call possibly maybe 100% map the entire proteomics, where do you see the most impact happening? Is it going to be in the drug discovery? Is it going to be in the biomarkers, personalized medicines? Is it the agriculture space or food science? Because there's rapidly, people are kind of build, recreating food as we know. So where do you see the most impact happening?
Thanks for this question. So, see, I would say that proteomics' major impact is essentially not to just confirm that this many genes exist in human, right? Because we already know that 20,500 genes are there, and that many proteins are going to be present, and only thing matter of finding those proteins. But I think what is most important here, how proteins get alternatively spliced forms, they make the post-translational modified forms with the different type of phosphorylation, ubiquitination, glycosylation. It means we are not looking at 20,000 number, we are looking at actually a million number. So even this proteome project, which will get completed soon, is still I think is going to open up the new dimension to look at characterize all the proteoforms, all the functional protein forms, for which the numbers could be millions, right? So I don't see that goal is getting completed in very near future, right? In China, there is a big project has started, PiHub, and where they have also involved international scientists. I represent India in that council for the PiHub, and that is very well thought of project of 30 years, where idea will be not only to just look at identification and quantification of all the protein, but also protein how they alter longitudinally in kind of, if you really want to look at the wellness, right, of individual, think about the switch point. Somebody is, very healthy, but suddenly some proteins start getting changed and that results into cancer, or that switch changes to diabetes or hypertension. So how that switch happens, right? Which is the time point we can find out that switch, and what are those factors which are driving that switch. Likewise, the goals will also be to then, can we now reverse those? If we can find the right switch, then can we try to reverse those switch? And before even disease get manifested, can we even try to reverse that disease and control that? Right? That will be, I think, the really cool if that happens. So the major impact for this field will be, especially when we are doing human research, will be in the human health, and it's already started making a lot of impact. I think now earlier all the treatments were very blinded, but now things are becoming more and more precision medicine based, and we are much more focused in our looking at judgments. So this is going to really impact a lot on human health. Also, it is going to help us looking at better effective drugs toward the therapy of different diseases. But the knowledge and the workflow what we are generating is actually equally applicable for the other model system as well, and therefore the agriculture and plants and all of that is going to be equally benefited with these technologies. So I think it's going to be very visible impact easily in the agriculture and plant, given that we can manipulate them easily. But in the human, I think it will take some time because of our restrictions on the ethics and the research for human. We know those switch point of the proteins which were involved in the changing from the fetal to the adult hemoglobin in the sickle cell, and now we are trying to utilize those gene knowledge and trying to edit those in the CRISPR. So again, this is where I think both the technologies and technology in general will help the human health and more toward the precision medicine. So this is one of the national facility we have established for the proteomics research and training. The one instrument here, this is the chromatography part, where, as I mentioned, you can take these individual samples and their peptides, and you can move those in this auto samplers. And from here, then those are going to pass through from this channel, and they will reach to this type of column which has the chemistry to bind the peptides or the protein. And then using these organic solvent, then based on their amino acid properties of hydrophobicity, then they are getting separated further. And here you can see like this is the zoom in panel where you can show these liquid form getting ionized, and these ions are going to go further inside this particular black box, which is having different type of mass analyzers like quadrupole, orbitrap, ion trap. So they are going to further separate based on mass and charge ratio. This is another orbitrap instrument, which is we are using for metabolomics. All the metabolites of human bodies, can we try to separate those and look at they come from nucleic acid or lipids or the proteins, and then we analyze them using different type of chromatography, UHPLC and the orbitrap. Typically, when I say the proteomics, as you can see on the screen, but faint, we get this kind of bushes type of spectra, which shows us the chromatogram of the peptide separation, which then we have to further search again the databases. And I'll talk about databases, why they're important when I go back. And this is another instrument which is known as the triple quadrupole mass spectrometer. Once you have identified and discovered the biomarker from the discovery work, you want to validate them on the large number of patients. So then you want to develop a test which is very fast and looking at very narrow number of protein, maybe 5 protein or 3 protein panel. So then we can use this instrument, which is the having the triple quadruple setup and UHPLC, and then that could be utilized for looking at targeted proteomics and metabolomics. And once, let's say, we have identified the proteins, we want to look at this protein binds to which drug or which inhibitor can control them. So we use different technology, which is one of this is known as the BLI, bioluminescent interferometry. And here we can take maybe 96 drugs and screen against the protein and trying to find out protein is binding with what kind of affinity with these samples. So we have this protein microarray scanner. So this is a very different instrument platform, like unlike the mass spec which you saw, which was we are taking the sample directly injecting it in the instrument and trying to look for all screening all the protein and peptides from those. In this case, we already have on the chip, we print all the protein and then we try to do the screening of these biospecimen and then we look for these signals. So for example, what you see on the screen, these are some chip spots which are showing the signal intensities. And then we do lot of, these are almost, I'd say 40,000 spots on a small chip. And this contains almost 19,000 of the human protein along with their duplicates. So then we try to look at their entire screening of the entire chip. So this is the chips, like this are just glass slides, which for anybody normally it will look like just a cell biology normal slide. But on these, we actually print all the proteins of a given organism, right? Especially you can just see here. So this is having almost 40,000 dots or the spots of human protein. So then if you have the even few microliter of the blood or plasma samples, you can screen those across on these chips and then look at their reactivity against these antigens, right? In the same way, we have actually done some work on the malaria for the looking at the P falciparum and P vivax chip, where we have printed all the antigens from falciparum and vivax. We have also done on the SARS-CoV-2 research. We had all the peptides of the virus, and we tried to look at their immune reactivity and which domains are becoming more important for screening. Since I'm in the lab, I can just show you that these biological sample, as we talked about central dogma of life, these proteins degrade very fast. So do you really want to make sure that they are protected in minus 80 kind of degree temperature? And you don't want the RNA or the protein to get degraded. So RNA and protein work is much more challenging as compared to the DNA or genomics, which is more stable even on room temperature. Here we have to really maintain a lot of things to ensure that the stability is maintained and there is no degradation happening. Yeah. So this is just kind of the broad overview of the lab, and we have of course several gadgets, including some of the NGS platforms which can do the more targeted sequencing. We have some immunoassay platform which can take few antibodies and look at their immune panel and screen those. So we have a variety of technology, and given the timing is short, I'm not taking to our facilities, which is having much more elaborate setup.
Really appreciate you running me through the your lab, which I think is so very awesome. Because you spoke about the China hub, the Pi hub, and here you are in India. I mean, doing the same. I mean, which I think is so very excellent, and you're forefronting proteomics here in India. So, which is very interesting. Congratulations, and more power to you and your team for doing that. You ran us through the lab. It'll be great if you could speak about maybe some of the work updates and what innovations has emerged from your lab. Will be great if you could talk about that.
So I would say like I'll split into two parts. One is the clinical aspect. Second is more computational aspect. So, clinical aspect, we are working on two major problems. One is the uh, malaria and the COVID-19, like infectious disease kind of projects. One thing which we realized that accurate diagnosis of malaria, while it was already there for falciparum, but other pathogen, which is the vivax, for that the diagnosis was not possible because the all the tests were based on the falciparum. So, and over the time, currently the major patient of malaria, they're actually from the vivax. So we did very detailed analysis for biomarkers from the blood samples of these patients from Mumbai and Kolkata. We had Bikaner, Rajasthan. We had Goa. Many populations we looked across from different hospitals, and we have now come up with the signature of three protein and their peptides, which are very unique in the vivax as compared to the falciparum. And through a government funding of MHRD, uh, we are working on a kit with a company to develop the RDTs, which can be distinguishing the falciparum with the vivax and also malaria with the dengue. Since during the COVID-19, then this problem came, and our lab was collaborating with many clinical hospitals of the infectious disease. Those clinicians requested us, can we look at the COVID-19 as well? And then we got certified to do research in that time for COVID-19. And one major contribution which we made was essentially we developed a assay which can help in to look at if individual is moving from the mild to the severe form of disease. On the brain tumors and the cancer side, there are two major initiatives we took. So in US with the Joe Biden's initiative, there was a project known as the ICPC, International Cancer Proteo Genome Consortium, and that was a part of their Cancer Moonshot project. They wanted international community to also participate along with the US NIH, National Institute of Health. I from IIT Bombay, we made a consortium and we signed the MOU to India to be part of the Cancer Moonshot project. So we took a lot of these brain tumor patients' blood sample, and idea was if the cancer in our body, the cancer cells are there, they will be suspected as the foreign cell in our body because in the way rapidly they are dividing, body's normal immune system after some time it starts sensing this is not normal. It is doing something very fishy. So then they try to raise some antibodies and try to combat that reaction. So while they're our own body still, they're not like virus, but it's still body's immune system is trying to control that. And in the process, they raise antibodies, which are known as autoantibody. It means our own antibodies are raised to control our own proteins from these tumor cells, right? So this thing happens much ahead of time in our body, even before the disease get manifest. Disease becomes very like late, maybe 10, 15 years later on. Only somebody is diagnosed as a cancer patient. But these changes start happening in our body much ahead of time, right? So can we look at the blood signature and try to look at these autoantibodies? These antibodies are much more stable entity as compared to protein. So then using the protein microarray, we try to look at these autoantibody response in different cancer patients. So this is something which is already being in the field, but I wanted to apply that more to the brain tumors, looking at in complex disease like this, uh, where we could find a set of four protein, which was indicative of lower to higher progression of the disease from very early stage using the blood samples. So this is something which is a very unique signature we have found. Additionally, we have also looked at in meningioma other type. This was what I talked to you about the glioma tumor, which is from the glial cell. Other is meningioma from meninges of the brain. And we have developed a test using the targeted proteomics, which is for 13 peptide panel, which can distinguish the low grade and high grade. So that is something which is very first time, where now we can utilize the targeted proteomics and use that knowledge to distinguish the low and high grade of the meningioma. And similarly, we are working and contributed a lot on pituitary tumor and the medulloblastoma, pediatric tumor, different type of things. So these are kind of a lot of clinical work which we are doing to generate the new knowledge of the Indian patient samples. Very recently, from the Indian Council of Medical Research funding, we have started a project at Northeast from different hospitals of Assam and Mizoram to look at colorectal cancer because it's very prevalent over there and what kind of protein and gene signature which is very unique over there. So that's something which we are working on. But while doing so, like in this process of looking at different type of brain tumor, we always felt there was a challenge of looking at the normal, like what to compare with, right? So most of the time we look at the peritumoral region, which means adjacent areas of the tumor, which will be normal, and try to compare that. But that often may have even confounding some tumor cell as well. So there was a need always to look at what should be the right control for the brain. Then we realized that given the normal brain proteome is not deciphered completely, and we do not have knowledge about even all the normal brain protein. So whenever we are looking at meningioma, we have to look at the arachnoid, dura mater, different regions of the brain. So then we thought, why not we look at systematically, first of all, what is the normal protein profile in all our brain region and sub-region. So then, as a part of the Human Brain Proteome project, Dr. Enevi from Spain and my team from India, including the enhanced team from Bangalore, we tried to come up with first do systematic screening of the normal brain of Indian individuals and the Spanish individuals, and look at what the protein expression level is in the different region and sub-region of the brain. So that was something which we did because we are always having the issues of looking at controls. So this is something which turned out to be quite big for the community because this is the first time which human brain proteome as well as the human brain phosphoproteome we were able to decipher. And then we made this database BrainProt, which was having the largest number of brain diseases and their entire detail of all the proteins. This database has been utilized very large number of researchers worldwide and very useful for the people to know the protein expression level across different diseases in the brain and different subregions of the brain. Additionally, what is also important now that we are looking at using machine learning, how to uh, use this knowledge and try to come up with the different panels which are the best biomarkers for different type of brain tumor. That's where a lot of undergrads from IT Bombay are involved, and they are trying to look at different aspects of it.
Couple of things, Professor. I think, I mean, you spoke about, the India government. What's the support system here in India? Because, I think the government is kind of pushing the Make in India agenda and trying to support its scientists, its researchers. What's the support system from government, from industry, and what are the biggest challenges of translating these proteomics findings from your lab to clinical diagnostic? And will also be great if you could also speak about, big data in proteomics, because, this generates like a whole lot of data. So what's the role of artificial intelligence in shaping the way we analyze and interpret these complex data?
So let me first take the big data part because I was talking to you about the brain plot, right? And that is again one of the way of looking at big data, which we try to put in the database. And so when we look at the brain plot, we had a variety of data sets from the proteomics and transcriptomics, and also we looked at these proteins. So we made different domains of the brain plot from the interhemispheric brain proteome map, or known as IBPM, to brain disease transcriptome map, TM, as well as the BDPM for the proteome map. And we also made one thing which was known as brain disease drug finder, BDDF, to look at all the brain diseases and which are the drug which can be already known available. Then human brain disease atlas we made, and we have also made the brain disease marker curator. So we did a lot of this thing from big data, but then we realized that lot of time and effort from the industry gets utilized or wasted in some extent to find out which proteins can be taken as a drug target. So, can we utilize some of the artificial intelligence based approaches and machine learning based approaches to narrow down those leads, right? So, we look at additional initiative after the brain plot, we try to further build for our brain drug finder, and we developed more recently about the drug plot AI, which is looking at the uh, druggable proteins and their identification with this computational pipeline which we have made. So we actually took all the 20,273 reviewed protein, and we took so we had there are almost 2,623 approved drugs, and what we did new was we used the maximum number of features which can distinguish these protein features, or almost 183 features, and we developed the novel partition ensemble classifiers to then look at using different type of machine learning program, for example, using the XGBoost or looking at random forest, build a model and try to predict the druggability of the protein. And then we started looking at the FDA data from the US government and look at how many of those proteins are actually turning out to be approved in the clinical trial and how many of them are getting failed. And that has really helped us quite a bit, that we are almost reaching 90% or close in making the prediction. And as the data is coming, then we are confirming though that is actually true prediction. So again, machine learning and big data is going to play a very important role to make our choices more smart and reduce a lot of wastage which happens, right? Because a lot of time, the industries put a lot of dollar value to look for a candidate in the clinical trial, and then at the end they get failed and they don't go to the final rounds. So can we at least make some smarter choice and the prediction so that they can narrow the choice, and then only those can be taken for the experimental part. So big data, whether we talk about proteomics or we talk about transcriptomics or genomics, all of them are generating big data, right? A simple project when we do, we have 200 file, at least 200 samples. Each file is almost 2 to 3 terabyte, and then now you think about how you are going to analyze the data, right? So it's a huge amount of data being generated, and it takes a lot of workforce and intellectual time to make sense of that data. And that's where I think AIs are going to play a very important role. It started already playing important role. We have now the AlphaFold, right, which is one of the game changer for the structural proteins. And based on the AlphaFold, we have to come up with actually another software, which we recently published in the very top journal of MCP, Molecular Proteomics, which has come just 2 days back, which is known as AlphaCross XL. So in the proteomics field, one goal is also to look at the protein which are like, for example, on the cancer cell, there are some protein which are showing the conformational changes. So can we identify those conformation change using the cross links, using mass spec? So, and how to analyze that data. So we have come up with a new software along with the collaboration from UCSF, University of California, San Francisco, where I did my sabbatical a year back. And then the Thermo Fisher from the California, they were also part of it, along with the many big teams. So then we came up with the workflow and the new software with Python-based programming, which can take this kind of crossing mass spec data. And then in the backend, it also uses AlphaFold to look at the structural prediction and then try to make prediction that this particular cross-link of mass spec goes where in the protein structure. So again, this is again very fast way of analyzing and locating and structurally elucidating the data, and that is going to be available to public now very soon because the manuscript already published. So again, you can see like in the different ways of we and others are now trying to utilize these resources and trying to accelerate the pace of making the right choices more, right? And still, I don't think they will rule out the validation part and the experimental part. We have to still do that, but at least it will reduce the choices, and we have to then work on limited choices to do the experiment and then eliminate those out.
Right. Professor, you spoke about this new model that you built, which is Cross XL, which is leveraging AlphaFold, and AlphaFold was a game changer of sorts. I mean, understanding, the protein structure, a folding problem. Now, AI is playing like a huge role, at this point in time, in all fields, and specifically in healthcare. This project that you're doing, I mean, how do you think it's going to play a role specifically in maybe understanding and reversing some of these neurological disorders? And because neurological disorders is the biggest problem, and at this point in time, I think the brain is like almost like a black box. It's like almost like AI. We don't know anything about it. With this understanding of the brain proteomics, first, maybe it'll be great if you kind of maybe give us like a lowdown on how do you capture the proteins from the brain, because we have like a basic understanding of the brain, through which we have invasive, non-invasive approaches of kind of mapping the human brains, and the various approaches like the human connectomics projects, and there's the organoids, which through your single cell you kind of create a pluripotent stem cell and create a brain organoid. These are like the various approaches, organoids, human connectomics projects, then these invasive and non-invasive ways, the EAGs and brain computer interfaces. How do you kind of approach the capturing these data, your proteins from the brain, and B, through these models that the model that you built, you're talking about AlphaCross XL, what would be what can we expect from it, maybe in the near term, and maybe in the future?
Right. And I think it is very broad question to answer, but I would say like these are uh, small tools which we are creating, right, to accelerate our understanding of brain and other diseases for that matter. Specifically for the brain disease which you asked, we actually took the cadaver, the patient who passed in the accident, right? Their brain samples. These are actual brain tissues from which we extracted the protein of different regions and the sub-regions of different hemispheres. In this case, we actually took the brain tissue of different regions. We extracted the proteome out of it. ran in the mass spectrometer, generated all the data, look at their proteome and phosphoproteome, and put them in a database. Now, this database is publicly available for the researcher. They want to work on the brain tumors or they want to work on the neurodegenerative like Alzheimer and Parkinson. Everybody can use the resource. So we are creating the resources for the community to accelerate, right? Not only individual our own research, but actually for the community kind of resource. Likewise, AlphaCross Excel is a community resource. People who are doing the cross-linking mass spectrometer and the proteomics of that, they can use this particular software to analyze the data at the unprecedented scale and the pace, and using the AlphaFold as the backend, which can now look for any protein structure for that matter. And therefore, now we are looking at for any disease for that matter. When you're doing the cross-linking mass spec, you can use our resource. Similarly, the drug plot AI, we have made as a public resource for the entire community to use to look at any protein and their possibility of predictability of the druggability of that protein. So again, many of these resources which I'm talking is actually going to help the community. And idea is all the researchers should start using these resources so that for many of the complex problems, including the neurodegenerative problems, we have the better answers. But again, like this is uh, these are just as I said, these are the small tools available, right? Which are going to connect the dots. These are not in itself is like going to give you right answer today tomorrow, but they are going to help the grow the community to address many of these challenging healthcare problems. If you want me to answer now the government part, or I can continue on this part.
That's an important spec of it. Will be great to hear your thoughts on that.
When I came to India and started talking about proteomics in 2009, proteomics was still quite new world that time, and but I took few years time to convince the the different committees and government, and finally I could see great support coming from government of India for different initiative I took, whether for the training programs or for research facilities, and in all the initiative they supported very well, and that has really created a lot of momentum. I have been also conducting many faculty training programs to train lot of faculty, 25 faculty every year. They come for few weeks in the lab and they do research and training. So again, this is kind of thing which is going to grow exponentially, and lot of new labs have been created in India which are now doing proteomics research. So those kind of government support has really helped both on the infrastructure setup side as well as on the training side. uh, I would say, like, if you think about the global pace, the way some of the big programs have been initiated in US and UK and China, I think that is something lacking. So that's what I think somewhere we have to convince and probably give our leadership a little bit more light on it, that in which way we can try to think about those setup, which is going to be not right away going to give us the output, but will be compounding in the over the years, right? Again, like the COVID vaccine did not come overnight, right? It was the several decades of the research and decades of the investment which has led us to help out in that time, right? So likewise, I think there is a need for some bigger common infrastructures in the country, where then big projects like this, when you think about thousands and lakhs of individual to be screened, individual lab and facilities like my cannot handle that kind of big initiatives alone. So we need to build those initiatives like China, the recent one which I mentioned to PiHub, where then there could be several hundred thousand individual screening and several hundred of scientists going to work on those centers and going to look at not only few master which I showed you, but maybe hundreds of instruments are there to work like the factory, right? So we have to work at that scale to then think about problems like cancer, problem like different type of neurodegenerative diseases, to be really trying to tackle that, especially a lot of India centric problems. So this is going to be, I think, something will be game changer over the year, but will require a lot of investment and bigger infrastructure setup from the government. That is something which I think will be important. But again, industry and government has brought in few schemes where they said if the industry is partnering to the projects, they will fund to the almost double the extent. So there are few schemes like that from the Ministry of Human Resource Development, MHRD, uh, where we had several industrial projects which came forward. But what industry looks for also, like industry looks for the quick turnaround, and they look for less investment but very quickly how to convert that in dollar values, right? So we have to think about more incubation centers where there is a more time for these technologies to get validated and then come to translation. So again, that is something which is, again, we need that pipeline to be created in the country, so that lot of basic research findings could be gone through a lot of validation, and then they're not lost into the publications and patent. They should be taken forward for the deployable products and the output in the kits form.
That's so very important, Professor, because I think countries like US and China are doing the right things. They investing a substantial amount of money in research, because when you research, you put the money. That's when you secure your future, and it's so very important. Because I think the even the investors here in India are very risk averse to funding the future and deep tech. I think that needs to change, and that stance also needs to change from the government itself. It needs to be bottoms up, because that's what possibly China is doing, getting it right. And I think that's the reason they are accelerating so rapidly and are today, maybe becoming like one of those global superpowers. I hope that India, the government, also takes a stance, and I hope that the investor community also understand that you need to invest in the future. Now, BrainProt, you use an omics based knowledge base. Then there's this human brain proteome project that leverages neuroproteomics. Will be great if you could kind of give us the difference, and would that possibly lead to maybe faster possibly diagnostic monitoring or neurological treatment, and collaboration. You spoke about the collaboration. Will be great if you could maybe elaborate on some of the collaborations that you're working towards.
That's right. So Human Proteome Organization (HUPO), where I'm have been part of the council several years, but currently I'm their member in the HPP Human Proteome projects. So they have initiatives for different type of diseases as well as different projects, including the brain project, right? So Human Brain Proteome project. So where idea is to look at all the normal proteins of the given organ or their role in the different diseases, right? And there are different teams from the world, they work together to with a common goal to look at that. Likewise, there are different teams working on just looking at the biomarker for maybe the Alzheimer disease, biomarker for Parkinson disease, biomarker for other type of cancer and disease like that. So there are many teams which are working on it. Um, Goal over there is to utilize this knowledge and put in one place so that while some teams are generating data, some teams are also validating them, some teams are validating to build the models with the computational tools and use that for making predictions as a panels. So there's actually big teams involved from the worldwide, and but this is not a common funded project. These are all scientists from different countries who are working towards the common goals, and then they have common interest, right? But again, as I said, if there is a kind of initiative from the government of any country to lead a specific disease project or kind of areas and invest into that, then there will be sizable intellectual mass which will be involved to really unravel that kind of problem, and that will be much more going to have incremental change to be seen in the population in the patients much more rapidly. But what all is happening with these initiatives is generating a lot of knowledge pool, which is going to help out the community in different times in different type of problems, right? But the goal is not immediately to have a kit coming out of it. That's not because there is no pipeline to have the industry taking these leads forward. And that's where, as we talked, there is a gap, to do that. But then the collaboration is the key, as you mentioned, in all of these areas. Like when I came back to India, the one thing which I realized while working at Harvard, collaboration was the key over there. For any project, people accept with very great humble way that we are not the expert of everything. So there is somebody expert of clinical side, somebody expert of the computational side. I'm expert on the biology side. Why not we, kind of make a collaboration? For each project, the team leaders, along with the postdocs and scientists, they go meet those kind of PIs and people talk each other and see can we make a collaboration. Therefore, every project has a different type of collaborative team, but very strong team which can actually look at the same problem from different angle, right? So that's something which I tried to build when I came back here, and I think I was very successful in thinking that way. And Mumbai definitely was very progressive. We have a lot of these clinicians, excellent clinicians from Tata Memorial and KEM and Kasturba Hospital. They were very progressive, they how busy they are, like when you go to their OPD and so many patients are waiting, but they still spend time in talking, and so many of them were co-supervising our students. So they spend a lot of time in thinking about research, which is really kind of really progressive and important, for these collaborations for the clinical research. Likewise, I think industry has shown interest for sure for our lab. they have seen a lot of promise, and a lot of investment has happened from Merck and Thermo Fisher and Shimadju and recently Bruker. All of these companies have made their center of excellences and provided a lot of funding to accelerate in several areas of research. But likewise, I think we need to have a lot more pharma sector to come forward as well, and the diagnostic companies to come forward as well, and try to help in taking these leads forward toward the quick translation of these things. So I think this is something which will be is already happening to some scale. Probably the overall vibes will change more rapidly, and there will be more stronger collaboration with the industry will happen. But clinical collaboration has been pretty strong in that way.
Right. Yeah, I really hope that the world moves more towards open source, because so far, everything is siloed, and I think maybe the the importance of collaboration was showed while COVID happened, and that was the first time I think we kind of figured out and built a vaccine in a extremely short period of time, only and only because the entire globe kind of collaborated and came together. I hope that we do that now, because there is so many problems, so many healthcare uh, which we could completely treat and reverse. I mean, if the world kind of collaborates. Wanted to get your thoughts also on the future of education. You've been the professor at IIT. What's the change that you're seeing? Because obviously there's artificial intelligence and people are getting afraid because of automation. But then there is this other side of people, people who are like, kind of keep on like pushing the conversation and leveraging artificial intelligence and thinking that, we can use artificial intelligence to augment us, so there's a couple of dynamics happening in the space of education. One is, I think the traditional model is being impacted by the e-learning or MOOCs, these massive open online courses, and you yourself have been part of it and created 10 national MOOCs, including India's first course on proteomics. So with this different dynamics, with these moves, massive open online courses which are pretty much free, how do you see the education being impacted because of that, and the role of artificial intelligence?
That's right. So I'll just split this thing into three parts. One is the classroom teaching at IIT Bombay. That has been extremely enriching, rewarding experience, uh, especially when we teach to undergrad students. These are engineering students. They come fresh from the JEE exam, right? First year students. And now at IIT Bombay, being very progressive, we have the biology also as one of the 101 course for them to take. So in addition to physics, chemistry, maths, right? So now teaching the biology and bioengineering to these undergrads are challenging and rewarding both way, because to get their interest, you need to really come and talk in their language, and to really make it very impactful, uh, you have to see, like, how you can utilize different type of technologies, right? So but what this investment is actually paying off, when I teach them in the first year, many of them actually are quite interested in the second year to come and do some internship and want to learn some projects. Eventually they start being part of our research teams and start contributing towards the big data analysis. Then they start bringing their programming skills and start looking at different machine learning to try to come up with the panels and the different biomarker signatures. So again, this is like, kind of the cycle where on one hand we try to teach them the fundamentals and bring their interest, and those who get interested, then they start bringing their skills and try to come into and become part of the teams. And likewise, the MOOCs has been extremely, I think, the big and wonderful initiative from Indian government, where now we have the Kusana Swayam, initially started from NPTEL from IT Madras. So MOOC has given the accessibility and reach of these knowledge from senior professors from different IITs and other big institutes to all over India, to different rural population, to every city. So in that way, like, I have been able to reach now thousands of students across India, maybe 40, 50,000 students I would have taught in MOOC courses. And what actually in turn, what we look at, these are online courses, but we still monitor their progress every week. Currently, my three MOOC courses are being offered. One on proteomics, one on proteogenomics, one on the proteins and gel-based proteomics. So in these courses, the students are doing the weekly assignment. We are also talking to them live every week, and then they will be writing exam. and these exams we make as tough as we make exam for IIT Bombay, our own students. So in some way, we are evaluating their quality despite being online to almost similar extent possible. And among those who are actually the toppers and who are really performing to the top, I feel like they are really good students and they need some incentive for being the top in the course. So I actually been offering them internship or some sort of exposure to the workshop or activity which we do at IIT Bombay. And many of them actually turned out to become the part of our team eventually. And what actually that helped because they're already well trained. They have taken my basic courses and they have gone to the training. And I have some successful names and stories in my lab. Students who came from MOOC courses, they joined in PhD programs here. They finished and they are now doing the research at Harvard and the kind of University of California in the top labs. And that gives a lot of motivation that okay, these kind of programs are really working out well because they are funneling down the best of the best from the our entire population, which is so therefore education is not very much now within the boundaries of the institute. It has really opened up to the entire community, and MOOCs is really playing fantastic role in that area. And I have been also like understand that since I teach in the Technology Institute, and many times teaching fundamental concepts of the clinical and biology becomes challenging to the engineering or the different audience, right? So therefore, I see that a role of technology is especially how to animate some concept, how to make the things little bit more lively for them, right? And I have had interest of even making some documentaries and some of the short films, which helps me a lot in my classes to really get them very captive to the audience and I pose a problem and ask them their interpretation, and that leads them to think about why we are going to talk about that fundamental technology. So again, education has to be thought in a way that the audience can really best absorb it. And so this technology audience is we have to treat them differently. And like right now, I'm teaching a proteomics course, but most of my students who are taking this course, these are undergrad students because I had to limit the number of students who can take the course because of the resource limitation. So most of them who quickly got the seat were all undergrads. So now I have made this more programming based course, more data analysis based course, and more open source software which we are utilizing in the course so that now they are getting tuned in their own language and they can bring their own expertise as they get comfortable in knowing how to analyze the proteomic data and their databases. They can then utilize their various AI tool to come up with the solution which was not possible earlier. So we try to expose them and then open them, keep them open to look into the unknown possibilities which they can explore. So this definitely like this part of our way of education and training what people at IIT Bombay, that how best we can nurture them so that they are exposed, but also they are independent to think about this bigger problem and their own way of providing solution to that.
Right, Professor, kudos to your approach in spreading this education. And you rightfully pointed out, and I think when there is some visual representation of what you're speaking, I think people tend to absorb it better. And I think that's what I kind of do with my podcast also. Because when I interview my guests, there are a lot of these complex subjects. A, I try kind of break it down. B, I try give some kind of visual representation so that it becomes like maybe more easily understandable. My last question to you: is there any advice that you would like to give young scientists or researchers who want to enter the field of proteomics and your Proteomica International Private Limited company? What comes next for your startup? And I want to put like three questions in one because this is my last question. Uh, will be great if you could like paint a picture of the world maybe in the next 10 years how this field of proteomics is gonna impact healthcare, and maybe it'll be great if you could kind of give details on which areas of healthcare you see it actually creating the largest impact. So three questions: advice to scientists, Proteomica International, your private limited, your startup, and proteomics and its impact in the next 10 years with specific details in the healthcare space.
That's right. So first of all, I would say that okay, currently the AI is a buzzword, right? Uh, the students who are actually venturing into the field, they should not just be blindly just running towards the new terms and the new field. The basics and fundamentals and the training in that area is first of all foremost important, because once you know the basics of your field, then only I think you can utilize different models to be smart to make predictions. But without that, you can't just be smart to make predictions. So you should still be, if you want to learn proteomics, you have to still learn the protein fundamentals. You have to still learn the technologies and their principles and how they operate and they generate data and how they analyze the data. Then only come to the eventual goal of looking at the big data and AI, right? So I'd still focus on the stronger basic training and the hands-on training, and then they can build their career further into the newer fields, right? Additionally, I think what is important, which is towards your third question as well, it's not only proteomics alone, but what is also people realize that in our body, all the changes are happening simultaneously, whether it's DNA, RNA, protein, or lipid or carbohydrate, they're all changing simultaneously. So, can we do multi-omics analysis of the same sample, not only just looking at one layer of information, but can we build the layers together and then make the prediction, which could be more system scale prediction? So even from the same sample, I showed you some other technology in the lab, which can do even lipidomics or metabolomics or other kind of omics investigation as well. So the next challenge is going to be how to integrate that data. For example, in one of the brain tumor project, recently we have on the same patient, we did all the genomics data and also proteomics. And now we're integrating as a proteogenomics, which is giving us some very, cool new signatures, which was very different than what is already known from the Caucasian population. So again, integrating data, building different layers, and systems approach, which will again bring a lot of computational power to then think more integrative approach and comprehensive approach of understanding disease is going to be the way forward. For so proteomics will play important role, but not the only molecule which will play answer to all the questions, right? And I think the major in this area will be precision medicine, where the things are happening on each individual, how we are looking at their all the molecules and trying to give them a better kind of diagnosis for the disease, or looking at the better treatment, or even prediction for their potential severity of the disease, right? So that's where for the cancer and other kind of other health diseases, this technologies and multi-omics is going to be very important, and started playing a lot of role already. Now you see like all the big medical centers, their teams have the different experts together. They sit on the tumor board and different kind of these discussion when they look at data from different dimensions before they are trying to come up with some solution. Yesterday night, I had a meeting from University of California, one of my collaborator. He's a pathologist and scientist over there who looks into brain tumor. And they were saying like, okay, within one room, and we had six pathologists to look at the molecular signatures and the omics signatures to even classify a patient should go to this group or this group. It needs a lot of discussion based on their signature. So again, this is where things are evolving. We have to not look at blindly, but very comprehensively every individual and their signatures to be much more accurate and more precise. And personalized medicine is where things are going to grow further, and proteomics will play a very important role in that. And now the last question is about Proteomica. So at Proteomica, we are trying to look at some of the crucial challenges and problems, especially towards maternal and child health is one of the key areas. Different type of neonatal disorders, different type of maternal health issues. How to look at that, and again, growing Indian population and lot of things for our population which has not been screened. Can we look at those early and those complications? Can we even think about the molecule which could be utilized as a patches and the belts which can make during the pregnancy if the women are wearing those patches? Those could help to predict those like the small tattoos and the watches to make those predictions and complications. That will be the kind of the future imagination for solving those problems. And we are looking at the baby steps toward that, and looking at the signatures of the protein and molecules like that. Additionally, we are also looking at understanding the wellness over the time period. Again, as I mentioned, some individual longitudinal sample, how that will work out. And then we are also looking at different type of cancer and ongoing projects and looking at biomarkers for that. Plus, we are also providing some of the services of the advanced pharma sector and advanced proteomics application, clinical application. So Proteomica in that way is comprehensively trying to utilize very different resources we have available and try to provide the answer to some many complex problems we have, along with different R&D which we are trying to build some new knowledge for the country in the crucial areas of wellness and the maternal and child health kind of space.
We are getting in a world where data is going to be the truth, because so far I think what we have done is the healthcare has been extremely reactive. I think we're going to go into a world which is very proactive, where we know every little thing. And you spoke about, that the body itself is so very complex, the DNA, the RNA, the proteins, the way it plays, binds together. I think, with multi-omics, maybe we will be able to understand our body better, and maybe we'll be able to go to a point where we'll be able to maybe go towards more towards precision medicine and precision healthcare. So kudos to you, your healthcare, your lab, and the entire team. Keep on doing what you're doing. And to my listeners, if you like what you see and hear, then please press the subscribe button. Until next time, see you guys. Bye bye. Thank you. Thank you, doctor. Really, really appreciate
So
this. Yeah.
much. Thank you.
Thank you.
Thanks for the initiative and wish you good luck for your channel and spreading the knowledge from different experts to the entire country. Thank you very much.